Welcome to our third video lecture on topic D3.2 Inheritance. This is our additional higher level content, so SL friends, you will not be responsible for this on assessments. Guiding questions continue to be: what patterns of inheritance exist in plants and animals, and what is the molecular basis of inheritance patterns? Our objectives for today: we're going to look at dihybrid crosses. In the previous video, we just did monohybrid crosses. We're also going to look at linked genes. We're going to define segregation and independent assortment and talk about how linked genes don't follow those rules. We're going to use notation, some special notation for linked genes. And then we're going to do some super fun statistics and look at how we can analyze the results of crosses using chi-squared. And then we're going to wrap up with a little look at gene mapping. Remember that prefix di means two, so dihybrid crosses, we're looking at two traits at the same time. In this example, we have seed color, yellow is dominant to green, and we also have seed texture, where smooth is dominant to wrinkled. We do like to use letters related to the dominant trait, so I'm going to pick big Y for yellow and little y for green, big S for smooth and little s for wrinkled. Of course, you don't have to use those letters. This is just common practice. So let's look at some genotypes of our parents in this example: homozygous yellow and wrinkled seeds. Homozygous is the same allele for the trait. Yellow is dominant, so I'm going to use big Y, big Y. Wrinkled was that recessive trait. I'm going to use little s, little s. So the genotype for these two traits together for this parent plant is big Y, little s, little s. The other parent plant is green and smooth seeds. Remember that green was recessive. I'm going to use little y, because again, we're homozygous. Smooth is going to be that dominant trait, big S, big S. Let's say that this is the parent that is contributing the pollen grains. It's the male plant. The pollen grains have sperm cells in them. Let's talk about what potential sperm cells we can make from big Y, little s, little s. Remember that in meiosis, we make these haploid gamete cells that only have one allele from each gene pair. And so if I'm looking at sperms that are in these pollen grains, they could have a big Y or a big Y. They're all going to have a big Y, right? They could have what kind of S? Well, a little s or a little s. They're only going to be able to have a little s. So for this genotype, the only possible sperm we can make are sperm with big Y's and little s's. Let's do the same thing for this parent plant that's making the eggs in the ovules of the plants. We have little y, big S, big S. We could make an egg cell that has, well, a little y and a big S. Those are the only options. That is not always the case, so be careful. But in this example, all of the sperm are going to be big Y, little s, and all of the eggs are going to be little y, big S. This is a super easy cross to make. My sperm, big Y, little s, meets that egg, little y, big S. Fertilization occurs. The genes from the sperm are going to be inserted into the egg to make that zygote, and we get a big Y, big S, little s. 100% of the offspring are going to be big Y, big S, little s. Their phenotype, 100% of the offspring will be yellow and smooth seeds. So far, so good? So far, so good. We're going to look at the same traits, but this problem's going to be a little bit messier. So still yellow seeds dominant to green seeds, that'll be our little y. Smooth seeds dominant to wrinkled seeds, our little s. Now our two parent plants are both heterozygous for both traits. That means that both of the parents have a genotype of big Y, big S, little s. Figuring out what potential eggs and sperm are possible from this giant genotype is way messier than what we just did. So we could have a sperm, let's do the sperm cells first, that has one big Y and a big S. So that's one possible combination of alleles for these traits that we could put into a sperm, but we could also have that big Y and a little s. What other combinations are possible? Well, maybe we have a little y and a big S. Also, we could have a little y and a little s. These are all the possible combinations of one allele from each genotype that we can put into eggs and sperm that we would make during meiosis. We're going to have the same combinations in the eggs. 1, 2, 3, 4 eggs. If you don't like that, guess and check what possible combinations there are. We can do FOIL just like we do in math class. We could have the firsts. The firsts of each pair are big Y and big S. I almost said little. Big Y, big S. We could have the outer, the big Y, and the little s. We could have our inners, the little y and the big S. We could have the lasts, the little y and the little s. Now our Punnett square is going to be, oh my goodness, 4 by 4, 16 giant big in the grid. And I don't have a ruler here on my drawing pad, so excuse these super wiggly lines. All right, let's do some fertilization. What if this sperm meets this egg? We're going to get big Y, big S, and a potential offspring. What if this egg meets this sperm? Big Y, big S, little s. You do not want to listen to me do all of these out loud, so I'm going to pause recording, fill in all these grids, and then restart. Feel free to pause if you want to do it yourself and then check your work. And here we have our completed Punnett square. So many possible genotypes. Let's look at the ratio of the genotypes. So we've definitely got one of these 16 is big Y, big S, big S. What else do we have? Looks like a big Y, little s. Are there any others of those? Why, yes, there is. That one. I have two, big Y, big S, little s. Homozygous for the color, heterozygous for the texture. How do you feel about me pausing the recording? And I will list these out because you don't want to listen to me count. So again, feel free to pause your video, count up, put together your ratio of genotypes, and then we'll check our work together in just a second. So hopefully you found also this ratio of genotypes. We have 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. Crazy messy. So many possible genotypes because there are so many possible ways to put these heterozygous genotypes together to make the eggs and the sperms, and then so many ways during fertilization to have new combinations in the offspring, which is why this kind of sexual reproduction is so amazing in terms of propagating genetic diversity in a species. Let's go ahead and figure out now our ratio of phenotypes. This is not quite as big a list. So phenotypes, we're going to look at the two dominant phenotypes for each trait first. So I'm looking for how many guys are going to be yellow and smooth seeds. So these guys will be yellow and smooth, and these guys will be yellow and smooth, and these guys will be yellow and smooth, and also these guys will be yellow and smooth. So 1 plus 2 is 3, plus 2 is 5, plus 4 is 9. 9 total yellow and smooth seeded plants. Let's figure out how many are going to be dominant yellow, but then recessive wrinkled. So here, dominant yellow wrinkled. This one will be yellow wrinkled. That's it. So 1 plus 2 makes 3 that will be yellow and wrinkled. Let's do the opposite. Let's do green and smooth. Green and smooth. Let's see. These guys will be green and smooth. These guys will also be green and smooth. 1 plus 2 makes 3. And that leaves us with one final phenotype to look at. The green, recessive, wrinkled, recessive, and there's only one. Highly recommend memorize this ratio of genotypes. No, this ratio of phenotypes. So sorry. Memorize this ratio of phenotypes, 9 to 3 to 3 to 1, 9 to 3 to 3 to 1. If you have a dihybrid cross where both parents are heterozygous for both traits, the ratio of phenotypes will always be 9 to 3 to 3 to 1, where 9 of the 16 offsprings should have both dominant traits. 3 out of the 16 should show the one dominant and one recessive, the opposite recessive and opposite dominant, and then 1 out of 16 will have the recessive trait for both. Statistically speaking, of course, this is all about predictions and expectations, not maybe what we actually always see. Good? All right, let's look at another dihybrid cross. We're going to put our pea plants away and look now instead at Drosophila melanogaster, also known as the fruit fly. Gray bodies are dominant to black bodies. I'm going to use big G and little g. Normal long wings, big N, are dominant to vestigial or short wings. That's going to be my little n. Notice here there's a different kind of notation. So we can, instead of using the capital letter for the dominant trait, instead we can choose the mutant trait and make that a lowercase. So little b for black body. Instead of using a big B for the dominant trait, now we throw a plus in there to show the wild type. So mutant versus wild type, recessive versus dominant, same ideas, just different ways of talking about it. We're going to use this way of denoting our dominant and recessive traits, but also these methods of notation might pop up in your future, so just know that they exist. Good? All right, so let's look at this cross. I have a fly homozygous dominant for both traits and a fly homozygous recessive for both traits. Let's do the genotypes of these parents. So homozygous dominant for both plants, both traits. Oh my gosh. I have peas on the brain still. Big G, big N, big N. Recessive for both traits, little g, little n, little n. So let's say this is the dad fly and this is the mom fly. Sperm cells can have a big G or a big G and a big N or a big N. So all the sperms are going to be big G, big N, little G. No, that's it. The eggs will be little g, little n. Those are the only possible combinations. So all of the offspring are going to be big G, big N, little n. 100% of the offspring will be heterozygous for both traits. That means 100% will have gray bodies and normal long wings. That was an easy one, right? Let's do another one. Let's do another example with Drosophila still, but now I'm going to do a test cross. Remember that test crosses? We're going to have one parent who is homozygous recessive, in this instance for both traits. We talked about test crosses a couple lectures ago. We use homozygous recessive because that will allow us to figure out if our dominant phenotypes are heterozygous or homozygous dominant genotypes. So again, test cross, we're using all the recessive traits. That lets us see interesting things about the dominant traits popping up in the population. So one of the parents, this one, the double mutant, test cross, homozygous recessive, this notation, we're actually going to use the little g, little n. The other parent is coming from the F1 generation. So the cross that we just did on the previous slide, we're going to pull one of those babies. Remember that they were all heterozygous. 100% were heterozygous for both traits. Big G, big N, little n. That's the same thing as this, just that different form of notation. Let's look at the ratios of genotypes and phenotypes that we would expect to see in the offspring. Let's make those F1 generation our picture here. So that's the mom. Let's make some eggs. So we could have an egg that is big G and big N, or we could have an egg that is big G and little n. But wait, we could also have an egg that's little g and big N. There's also the possibility for an egg that is little g, little n. What about the sperm? So here's the dad fly. He's little g, little n, which means that we could make sperm that is little g, little n. And that's it. There are no other possible combinations. So I only need to put one column in my Punnett square. I don't need to build it 4 by 4 like we did in that other dihybrid cross, because this is the only possible combinations of sperm. All the sperms will look like this. Let's do some fertilization. We get a big G, big N, little n. We could also have big, little, big, little, little. So our ratio of genotypes is going to be 1 to 1 to 1 to 1. That one, that one, that one, that one. Let's talk about phenotypes. This one is going to be gray and normal. A fourth of the offspring should be gray and normal. A fourth of the offspring should be gray with short wings. A fourth of our offspring should be black with normal wings. And a fourth of our offspring should be black with short wings. So we should have a 1 to 1 to 1 to 1 ratio. 1/4 of the offspring should be each of those combinations of traits. Hold on to that idea because we're going to look at what actually happened when we put those flies together. These are the numbers of flies that were actually born with those different combinations of traits. Instead of 1 to 1 to 1 to 1, we have like 10 to 10 to 2 to 2. I guess I would actually reduce down to 5 to 5 to 1 to 1-ish. Lots of rounding, right? Seriously, lots of rounding. But we definitely do not have a 1 to 1 to 1 to 1 ratio of these different traits. Instead, we had a lot of offspring that looked just like that mom. And we had a lot of offspring who looked just like the dad. And very few, relative to those numbers, very few had a mashup of the traits. A little bit of mom's gray and a little bit of dad's short wings. A very little bit of dad's black color and mom's normal sized wings. That's weird. And so our ratio of the phenotypes did not match our expectations. And scientists were looking at this and were like, what? That doesn't make any sense. Why is this happening? And it turns out that it's because those two genes, wing size and body color, are on the same chromosome. And so during meiosis, this chromosome went together into an egg or a sperm, which means that those traits were inherited together. We call these traits linked genes. Linked genes are traits that are coded for, have genes that are on the same chromosome. And interestingly, the more closely they are located on the chromosome, the more likely they are to be inherited together. Linked genes commonly inherited together because the sequences of DNA that code for those phenotypes are on the same chromosome. So super cool, right? So cool. That cross we just looked at that did not have the 1 to 1 to 1 to 1 ratio that we were expecting violates Mendel's laws of segregation and independent assortment. Mendel looked at those pea plant traits and they happened to not be on the same chromosomes. Maybe he picked them on purpose that way, maybe he just got lucky. But all the traits that he looked at did show segregation and independent assortment. The traits were inherited independently of each other because they were on different chromosomes. So let's say that I'm looking at this diploid cell that's about to go through meiosis to make some eggs or some sperm. During anaphase 1, we separate those homologous pairs of chromosomes. And so we have these first little sets of cells at the end of meiosis 1. And then these guys separate during anaphase 2. And we end up with some cells that are a big A, little b, little c, big D. Those guys are the same. Over here, little a, big B, big C, little d. So what if I switched up how I paired my pairs? So we just said that we could have big A, little b, little c, big D, or we could have little, big, big, little, right? What if I trade the order of these chromosomes? And so I put orange on this side and I put purple on this side because those chromosomes line up in their homologous pairs independently of each other. Kind of like independent assortment. So now when I go through meiosis 1, anaphase 1, I get these two haploid cells. Let's go ahead and go through meiosis 2, anaphase 2, and we could have a cell that is big A, big B, big C, little d, or we could have some cells, some eggs or sperm that are little, 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 big. Notice that sometimes we could have big A with little b or big A with big B. Those guys segregated separately, independently of each other. They assorted into eggs and sperm independently of each other. I could sometimes get this and this, or this and this. Or we could get little and big, or little and little. Those guys did follow the rules of segregation and independent assortment. But what about the B's and the C's? Notice here that little and little stayed together, and little stayed together, and those two big stayed together, those two big stayed together. Let's look at our C's and D's. Notice that I've got big and little, we have big and little, and we have big and little. Notice that we've got over here little and big, little and big, little and big. They got inherited together because they were next to each other on the same chromosome. We call those traits linkage groups. They are linked genes in the same linkage groups. They're inherited together because they're right next to each other on the same chromosome. One way that we can show gene linkage is with this special kind of notation. How do I know if genes are on the same chromosome or not? Well, these lines are kind of like the beginning of a chromosome. So I actually like to draw all the way around. I like to draw all the way around. And I'm like, here is one chromosome, and here is another chromosome, and this is a pair of homologous chromosomes. And so on one of the chromosomes, I have big A, and on the other chromosome, we have little a. And on the chromosome, we have a big B, and on the chromosome, we have a little b. This helps me see that on this chromosome, I have these two genes. These genes are linked because they are on the same chromosome, and they're probably going to get inherited together. These genes are also, well, I mean, they're the same genes. They're linked because they're on the same chromosome. So when I do a cross, I'm going to make some, let's say that this is the mom. I'm going to have some eggs that are big A, big B, and I'm going to have some eggs that get this chromosome that are little a and little b, and that's it. I don't have to do all the other things because that's it. Egg is going to get that chromosome or that chromosome. If egg gets this chromosome, then this is it. If egg gets this chromosome during meiosis, then this is it. Let's do the same thing with those sperm on the other side. So let's say that this is the dad making some sperm. This chromosome might end up in a sperm. Big A. Ooh, that's a little a, big B. Or this chromosome could end up in a sperm, and we could have little a, little b. And then we do our cross, and we get bigs and littles and bigs, bigs. Big little, little little, big little, little little, little little. Notice that these offspring have both dominant traits. This offspring looks a lot like dad. These look a lot like the mom. This is the only one that got a little bit different, which makes sense because we should see just little bits of differences from those parents with those linked genes. Most of the time, those traits are going to be inherited together. Except when crossing over occurs during prophase 1 of meiosis. What? I know, it's so exciting. So let's draw our chromosomes back. Let's say that I've got those two chromosomes, but during prophase 1, crossing over occurs. We form a chiasma right there, and then these two chunks get traded, and so now I'm going to have one chromosome that has the big A allele and the little B allele. And then I'm going to have another chromosome that has the little a and the big B alleles. Because these are combinations that are different from the parental combinations, we call those recombinants. These are recombinant genotypes, recombinant chromosomes. They were produced because of crossing over. And so now we have new collections, new combinations of phenotypes that are going to pop up in the offspring. this is a source of genetic variation in the species. It's very, very exciting. We call these recombinants, and they allow us to see those new combinations of traits popping up. Of course, crossing over is not going to occur exactly between those two genes on this chromosome every single time meiosis occurs. And so it's only every once in a while that we get these recombinants, these new combinations of traits. We saw that in the fruit flies. Remember that fruit fly cross where we thought we were going to have a 1-to-1 to 1-to-1 ratio, and instead we had a 5-to-5 to 1-to-1 ratio? A lot of rounding. There was like 1,000 of the baby flies that matched mom with the gray body and normal sized wings. There was about 1,000, again, lots of rounding, that had the black body and short wings. That was in the dad fly. And only about 1 out of 12, a fifth of either of those other traits, did we see recombination, where we had a gray body and short wings, or a black body and normal sized wings. These were those recombinant phenotypes that occurred because crossing over occurred. All of these offspring were born from those not crossed over chromosomes. So cool, right? Super fun. Genetics is amazing. You know what else is amazing? Math. Math is pretty amazing. We're going to do some math here to help us know when the results that we get from our genetic crosses are different enough from what we expected to see from our Punnett square for us to say, oh yeah, linked genes here. The test that we're going to use, the math that we're going to do, is the chi-squared test of statistical significance. The formula for it is here. We're going to do it in this box because I love a data table. This is going to help us determine if the differences that we're seeing in the offspring are statistically significantly different from what we expected to see in our Punnett squares. It was first done with pea plants by these two scientists. This guy's name, Reginald Punnett. I bet you can guess what he invented. And then this other guy, Bateson, his partner. His face looks a little bit grumpy, but this quote is so good. Treasure your exceptions. When there are none, the work gets so dull that no one cares to carry it further. He's saying when things are weird, when things don't go the way that you think they're going to go, that's when you spend all your time and effort trying to figure out why. And then all the amazing science happens. Back to those peas. So what they did was they took some homozygous parents. Purple is dominant to red and long is dominant to round. And both of the parents were homozygous for both traits. So we had big P, little, big L, little, 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 little. And then we cross those guys. And of course, that first generation, that F1 first filial generation is going to be big P, big L, little l, because the parents, that's what you're going to get. But then they did some self-fertilization. So they took these plants and they crossed them with themselves. So heterozygous for both crossed with heterozygous for both, basically with themselves. And then they looked at what they got. We're going to build our giant Punnett square to show what we are expecting to see in the offspring. I'm going to pause the video because it's going to be one of those 4x4, 16 big grids. If you want to also pause the video and do that Punnett square, do it. Otherwise, hold on just a second. And this is what our Punnett square looks like. And this is what our ratio of phenotypes looks like. We would expect to see 9 that show purple and long traits because those are the both dominants. We should see 3 out of the 16 are going to be purple and round. 3 out of the 16 are red and long, and then only 1 out of 16 should have both recessive phenotypes, the red and round. So they actually did this cross, and these were the results. This is what they observed when they carried out the cross. So there were 284 that were purple and long. There were 21 that were purple and round. there were 21 that were red and long, and 55 were red and round. Let's figure out what we would expect to see with our 9:3 to 3:1 ratio if I have 381 total plants. So 9 out of 16, 9 divided by 16 in my calculator is 0.5625. 0.5625 times 381 gives us 214 plants we would expect to see be purple and long, because 9 out of 16, because 3 and 3 and 1 and 9 is 16. So 9 out of 16 times 381, we expect 214 purple and long plants. Purple and round, 3 out of 16 ought to be purple and round. So 3 out of 16 times 381, 3 out of 16 is 0.1875. 0.1875 times 381 gives us, we should see 71 and a half, I'm going to round that to 71 offspring are purple and round. Red and long is also that 3 out of 16. 3 out of 16 is still 0.1875 times 381. We should have 71 offspring that are red and round. Ooh, red and long, sorry, red and round. We have 1 out of 16. So 1 out of 16 is 0.0625. 0.0625 times that 381, we're going to get 23.8. I'm going to round that to 24 offspring are expected to be red and round. And again, I added up all of the offspring. That's 381, which is why I'm using 381 here. And then I just took those fractions. 9 out of 16, 9 divided by 16 times 381. 9/16 of 381 is 214. So we expected 214, but we actually observed 284. I'm going to now, next part of chi-squared test of statistical significance, I'm going to subtract my observed and my expected. So observed minus expected. Observed minus expected. What's the difference? 214 minus 284 is negative 70. 71 minus 21 is 50. 71 minus 21 is still 50. And then 24 minus 55, 24, sorry, clicked the wrong button on my calculator. 24 minus 55 is negative 31. So far, so good? So far, so good. So now I'm going to square all those numbers. So negative 70 squared is 4,900. 50 squared is 2,500. 50 squared is 2,500. 31 squared, I have to do that in my calculator, is 961. Now I'm going to take all of those numbers and divide them by the expected. So 4,900 divided by 214, 4900 divided by 214 gives us 22.90 when we round. 2,500 divided by 71, sorry, is going to give us 35. 2,500 divided by 71 again is still 35. And then 961, our square of the difference divided by the expected 24 gives us 40. So now I'm going to add all of those up. 22.9, I'm going to round that to 23. 23 plus 35 plus 35 plus 40 gives us a grand total of 133. So this is my chi-squared value. What does that even mean? Well, now I have to go to this chi-square distribution table, and that's going to help me figure out if this is statistically significant or not. How this works. First, I need to figure out my degrees of freedom. Degrees of freedom. So I have 1, 2, 3, 4 possible different things that my plants could be. They could be this, or this, or this, or this. How many ors are there? How many freedoms do we have to be different things? Well, we have to be one of them, but there are 1, 2, 3 other things that we could be. There are 3 ors. The other way to think about this is you figure out how many different combinations there are. That's 4. Take 1 away, and we get 3. So we have 3 degrees of freedom. In the natural sciences, for us in bio, our p-value must be less than 0.05. These are my p-values. These p-values, p for probability. Whoa, I spelled probability wrong. So close. So a 0.05 p-value, 0.05 or smaller, means that there is a 0.05 chance, or if you like to think in percents, better, a 5% chance, or smaller, that the differences that we are seeing just happen kind of randomly, because sometimes random things happen. So again, in the natural sciences, our p-value must be 0.05 or smaller, 5% or less, for us to say that our differences, that our results are significantly different from the expected. We say that we can reject the null. Null means nothing. We reject that nothing changed. Over here, if I'm greater than that 0.05, we fail to reject the null. Basically, we're saying that not nothing happened, really nothing happened. Nothing changed. Nothing changed. Something changed. So now our degree of freedom number 3, I'm going to go across and run into my 0.05, because in the natural sciences, that's our golden number. So I'm going to call this my critical value, my F-crit, my critical value, a 7.81. My chi-square value needs to be greater than or equal to 7.81 in order for me to say that my p-value is less than 0.05 and my results are statistically significant. Friends, 133 is so much bigger than 7.81, and so therefore our p-value is much less than 5%. We are much less than 0.05. That means that our results are, in fact, statistically significant from the expected. Something happened. What happened? These genes are linked. It's a lot to think about. I promise we're going to do lots of these practice problems in class, so don't panic if you're like, what? I know, it's a lot. And one of the amazing things that came out of all of that math is that scientists were able to map genes on chromosomes. By looking at how frequently genes were inherited together, we were able to figure out how close they were to each other on chromosomes. So these guys, this is chromosome number 7 of humans. We call the short arm the P arm and the longer arm is the Q arm, the centromere in the middle. And of course, remember that there are also telomeres here at the ends. These genes are linked because they're pretty close to each other on a chromosome. We can actually look at gene mapping in databases that are free and open to the public. So if you're like, I love a good database, or if you come to class, I'm going to make you love a good database, here are some super fun ones that you can play with. So the NIH's National Library of Medicine, the NCBI National Center for Biotechnology Information, is free, and you just like plug in, I want to learn about cystic fibrosis, or I want to learn about ABO blood types. You can use this other one, gene cards. Both of these are amazing and fun. And I love especially this one because it is actually internationally collaborative. And so lots of different countries, lots of organizations have been working on gene sequencing and gene mapping, and then they shared their data so that scientists everywhere can have access to all the most amazing and complete data. And on that note, my friends, we made it to the end of this video lecture. We talked about those dihybrid crosses, looking at 2 traits at the same time, linked genes, genes on the same chromosome. Some traits will follow the rules of segregation and independent assortment that Mendel talked about. Linked genes don't because they're on the same chromosome. We talked about that fun notation for linked genes where we use the lines and then we put the letters like this. Feel free to draw those chromosomes and then think about how sometimes linked genes aren't linked anymore when crossing over occurs. We talked about chi-square, and then we talked just that tiny little bit about gene mapping. Great work today. It was a lot, I know.